We know a great deal about the molecules involved in this rhythm and how they interact with each other from examination of the model organism Drosophila melanogaster, commonly known as the fruit fly. While we share very little physical resemblance with a fly, we share remarkable similarities at the molecular and cellular level with them. Therefore, we can learn a great deal about how and why we sleep from studying these small and mighty flies. Our next speaker uses the fruit fly to untangle the relationships among circadian rhythms, sleep, and physiology, to understand sleep homeostasis, the how and why we sleep. Please welcome Howard Hughes medical investigator, the John Musser Professor of Neuroscience, and the director of the Penn Chronobiology Program at the Perelman School of Medicine at the University of Pennsylvania, Dr. Amita Seghal, who will talk about using a simple animal model to understand how and why we sleep.
Thank you, Margaret. And I'd like to start by thanking the organizers for inviting me here to give this talk. It's really an amazing and an unusual conference. It's a great experience to be here and also in this beautiful location. So thank you, and I really look forward to the rest of today and all of tomorrow. So you heard that this is what I'm going to be telling you about, using a simple animal model to understand how and why we sleep. And as you just heard, this simple animal model is what is commonly known as the fruit fly, and its biological name is Drosophila melanogaster. So what I'm going to do in my talk today is cover these three different topics that I have here. The first being, why a fly? Why are we choosing to work with this? The second, can we use the fly to determine how sleep is controlled? Like, what drives sleep across the lifespan, but also different molecules? What makes you fall asleep? What makes you feel sleepy? And then finally, can we use the fly to determine the function of sleep? In other words, why we sleep. So, why a fly? And for this, I'm going to have to go to circadian rhythms first, because sleep is a rhythm which occurs with a periodicity of about 24 hours. And this circadian rhythm is found in almost all organisms. And not the rhythm of sleep, but just rhythms in general, 24-hour rhythms are found in almost all organisms. And these rhythms, as I just alluded, are not just of sleep. So shown here on this 24-hour clock are some of the processes that in human show time of day specificity. In other words, they tend to occur at specific times of day, or they tend to not occur at specific times of day. And I would stress that this is just a subset of the processes that cycle over the course of the day. So you'll see here that we're talking about blood pressure, body temperature, melatonin secretion, GI function, and there's many, many more that are not on here. So for instance, your glucose is different at different times of day, your cholesterol is different at different times of day. Almost everything cycles. In fact, it's hard to find an aspect of physiology that doesn't on some level show circadian regulation. How are these rhythms, why should we care about these rhythms? And we should care about them is because when they're disrupted, and they can be disrupted by intrinsic factors, so you could have a genetic mutation that gives you aberrant rhythms. They change with aging, but they can also be disrupted by extrinsic factors. So we heard about shift work that was alluded to earlier today, and that is a huge disruptor of circadian rhythms. If you travel a lot, you've experienced jet lag, and you know that your rhythms get really messed up when you do that. Even eating at the wrong time of day will affect your circadian rhythms. And when these rhythms are disrupted, you have a susceptibility to neurological and psychiatric disorders. This includes neurodegenerative disorders like Alzheimer's. And you also are more susceptible to metabolic cardiovascular disease. I'll point out here, as long as I'm talking about disrupted rhythms and things like shift work, is that often with shift work, you know that a lot of bad things happen, and it's not clear whether it's because of the sleep loss or because of the disrupted rhythm, okay? So teasing those apart can be a little bit tricky. So how are these rhythms generated? And these rhythms are generated by clocks that are within us. Okay. And these clocks are found in almost all body tissues, in the brain and the liver. So, you know, your rhythm of glucose and cholesterol is driven by a clock in the liver, shown here. Rhythm of insulin is because of a clock in the pancreas. Now, we knew that all these tissues contain clocks only after we figured out what the clock was, right? Because before that, like before we had any idea of the molecular nature of a clock, people thought that there must be a clock in the brain that just drives everything. And it was only after the molecular basis of the clock was found that we realized that these clocks are really everywhere. So the question then was, how do you generate a clock? What is the molecular basis of the clock? And this is really where, for purposes of our field, the fruit fly became really important. Because it was in flies that this basis of the clock, the mechanism by which you generate a clock, was figured out. How do you measure rhythms in Drosophila? So there are different ways you can measure rhythms because flies like us have rhythms in several different processes. But perhaps the easiest and the most used method is to monitor locomotor activity. You put individual flies in glass tubes, such as the one shown here. You project an infrared beam into the tube. The fly can't see the infrared light, but when it's active, it marches back and forth and deflects the beam, and that's picked up by a computer. And you generate an activity record that looks something like this. And so the shaded areas here are activity, and the data, so this particular fly was monitored over a 10-day period, and the data are double plotted. So this day is plotted here, and then again at the beginning of the next, and that allows you to see the transitions from one day to the next. And you'll see here that so there is periods of activity and then periods of rest. So, so you can measure circadian rhythms. Now, how do you address how they are generated? And this was attempted first by Ron Kanapka, who was a graduate student in the lab of Seymour Benzer at Caltech. And what he did was to take advantage of this very powerful approach in flies, which is called forward genetics. And basically what you do is you take animals, you treat them with a mutagen, so you introduce mutations randomly across genes, and then you assay all these different flies that now have mutations in different genes and ask which one affects your process of interest, in this case, circadian rhythms. And then you go in and figure out what gene was disrupted to cause that effect. So Ron Kanapka was fortunate enough to find every kind of mutation you might want. that would affect circadian rhythms. So back in those days, this was published in 1971. There were no computers. And so, but you could monitor activity using a chart recorder. Okay, it was basically like a needle on a chart, and it would give you a deflection every time the fly moved. So the process was actually very similar. And in some ways, I like these data better than the computer-generated ones, because every little vertical tick here is the movement of the fly. So you can just, looking at this, tell exactly what's going on. The data are the same as before, except now each line is 24 hours, and the ticks are activity. So the wild-type animal has a period of 24 hours. It's active at about the same time every day. The mutation, one of the fly lines, or fly families, had a 19-hour period, and you can see that here in that activity each day begins and ends earlier than the day before. Okay, they're running with a short period. And then he got animals that had a long period of 28 hours, and you see that here in that, activity each day is starting and ending later than the day before. And he got animals that were arrhythmic. They had no rhythm at all. So they're just randomly active throughout the day. Now, this was 1971. In those days, there was no molecular biology. So when I just said, when you've got your mutant fly, then you go in and figure out which gene was disrupted, that could not be done in the 70s, okay? So it wasn't until the mid-80s that the period gene was identified, and that turned out to be the gene that is mutated in all of these mutants. So it's the same gene with different mutations can give you these different effects. Okay, just the same gene. And so it was identified, as I said, in the 80s, and it was done in the labs of Michael Young at Rockefeller, who was one of your speakers last year, and in the lab of the joint labs of Jeff Hall and Michael Rosbash at Brandeis University. Now, fast forward 20 years from here, and this is when I was a postdoc in Michael Young's laboratory and did a screen for new mutants that might affect circadian rhythms. And we got my fellow coworker and postdoc, Jeff Price, and I found this mutation that's called timeless, or that we called timeless. And I hope you can see here why we do, because these flies, when you release them in constant conditions where there are no external cues, they have no rhythm whatsoever. So we isolated the timeless gene. Some of this work took place after I had already moved on to start my own lab at the University of Pennsylvania. And we came up with the clock model based upon some findings with period or per that Michael Rosbash had previously reported. But basically, these two genes function in a loop. They make RNA, as all genes do, and the RNA makes protein. But these things happen at specific times of day. And the protein goes back and turns off the gene, okay? And then the protein at some point goes away, and then the gene starts getting made again. So you have this loop that takes 24 hours to complete and is the molecular basis of these rhythms that you see. And so very simply put, you can think about the activity of these proteins as telling the animal what time of day it is. So this mechanism, and most of the genes, I'm simplifying it here, so only showing these two, but most of the genes that were found in the fly to be part of this clock turned out to be conserved in mammals. And we now know are mutated in human circadian disorders. And this led to the Nobel Prize in Physiology or Medicine, 2017, being awarded to these three individuals, my postdoc mentor, Michael Young, and Michael Rosbash and Jeff Hall, the people who had initially identified the period gene and like I said, then, together with period, we had timeless that helped us put the clock together. So in this, poster that was put out by the Nobel Assembly, you see, of course, the Earth rotating around its axis, which is why we have rhythms, examples of a rhythm, opening and closing of the leaves of a plant. And then here are our two friends, the period and the timeless genes. So, I was encouraged to share personal stories in the course of this talk. So here is where I will share one, and this is in particular for the trainees among you all. When I isolated, when Jeff Price and I isolated the timeless mutant, I wasn't sure whether I really believed our findings, okay? And for a couple of reasons. For one thing, people were even skeptical whether or not we really had a mutant. And I don't blame them for being skeptical, because I myself was so insecure, because I kept thinking, it's been more than 20 years since the Per mutants were found. What are the chances that there really is something else out there that, why wasn't it that somebody else found it before us? So is this real? And then the other thing was that I always had very bad hands. I felt like I did science a huge favor when I stopped working at the bench myself. And so I never generated pretty data. I always felt, insecure about how it looked. And so that was another issue where I was like, I don't know, this is not looking good. So imagine my surprise when I got to go to Stockholm for these ceremonies as Mike Young's guest, and I saw on this poster my data. Okay, and on the one hand, I was embarrassed by it because these actually are pretty ugly looking data. But on the other hand, of course, I got a huge thrill out of it. And the reason I share this story is because I think all of us who do science are always a little nervous about what we find, right? Because we're doing things that have never been done before, carving a new way. And so it's natural to have some, lack of confidence in whether or not this is really going to pan out. And I think the lesson is that, stick with it and, follow your gut and do what you think is right. And you never know when what you're doing today will turn out to be important tomorrow or 25 years from now. Okay, so as I said, this fly mechanism was very well conserved, so the fly turned out to be this amazing model for figuring out molecular basis of circadian rhythms. So we asked, can we use this to study sleep? Then we can do all kinds of experimental manipulations. And, as it was mentioned before, sleep is still a relatively new field in terms of mechanistic research that's being done on it. And this was even more so true 20, 25 years ago, where hardly anything was known about the molecular nature of sleep. And so there was this sense that, we needed to jumpstart the field in some way and take more genetic approaches, more molecular approaches. And we knew that in the fly, at least for circadian timing, which is an aspect of sleep, you could take these approaches. So the question is, can you do it for sleep? But what we needed to do was first see whether these bouts of rest that are in between activity bouts in flies, whether that is sleep. Are they just resting, or are they actually sleeping? And as Margaret alluded to, it's because there are many things to sleep that are beyond its circadian control. Sleep is its own thing. So typically, the timing of sleep is controlled by the clock. You sleep at night and you're awake during the day. But you all will have noticed that if you haven't slept well at night, or not slept at all, then the next day you want to sleep, even though your circadian clock and the alarm clock are telling you to wake up. And that is an example of the homeostatic control that Margaret talked about, that we all have the need to sleep. You heard Dr. Stickgold talk about this too. We all have the need to sleep. We need a certain amount of sleep. When we don't get it, we have to make up for the sleep we lost. Okay, so the homeostatic control is really critical. So it turns out that flies have a homeostatic need for that rest they have. Okay, yep. And could you please play the video? So this is, I apologize for the quality of this. It was taken many years ago with the shoddy camera, probably the first version of the cell phone camera. But I think it'll make its point, which is what you're looking at here is a bottle of flies in the morning. These flies slept at night, and they're active. This is their active time. These guys on the left, I assure you, are not dead. They are undergoing their sleep rebound because they were kept awake at night. Okay? And so, after a few minutes, they will, I don't know if it's within this video or not, they will wake up and start moving around. Okay, so they show a homeostatic control. And in fact, there are many aspects of fly sleep that meet the criteria, behavioral criteria that have been proposed for sleep over the years. And all of this was shown by Joan Hendricks, who was a sleep researcher who came to my lab to do a sabbatical. So Joan Hendricks was she was a VMD PhD. She was a vet who was researching sleep apnea in bulldogs, because that entire species, bulldogs, they have sleep apnea. So she was researching sleep apnea in bulldogs, and she came to my lab to learn molecular biology. And this was at a time when we were hearing a lot from a lot of organizations, the NIH, the Army, the Air Force, about how we needed to get the sleep field going to do some of the stuff that the circadian field had done. So Joan took it upon herself to develop the fly sleep model in my lab. And so basically, we already knew it's a reversible state of behavioral quiescence. It's consolidated by the circadian clock. I just showed you about the homeostatic control. And the sleep in flies is associated with an increased arousal threshold. What that means is that when the animals are sleeping, they are less sensitive to sensory stimulation. And that's a hallmark of sleep. You need a stronger stimulus to get the animal to respond. Now, there are other features of sleep that are reminiscent of human sleep. You can just read them here. I won't say them. But those on top are the important ones. So we developed this fly model for sleep, and it was simultaneously developed by Paul Shaw in Giulio Tononi's lab. But I want to stress that we were not the first to think about using such small animal models for sleep. Because Irene Tobler, who I really think was a pioneer in Switzerland back in the 80s, had started to ask whether you could use small animal models. And the model she used was the cockroach, where she tried to deprive it of sleep or its rest with forced locomotion. And she made this statement about how immobility in invertebrate could be similar to sleep in vertebrates. So we do indeed measure sleep in the fly routinely by measuring activity, right? So it is based on immobility. We know from work, some very heroic experiments done by others, such as Ralph Greenspan, that there are electrophysiological correlates of the sleep state in flies. But it's not practical to stick electrodes in the fly brain for every measurement, sleep measurement, okay? So we typically just use activity, and it's not any immobility, it is a 5-minute or more period of immobility that qualifies as sleep in the fly. And I will just note that even in humans these days, activity, so actigraphy, which basically is monitoring your activity over, 24 hours over a period of days or weeks, is being increasingly used in sleep medicine to monitor sleep. So the fly model we developed for sleep ended up becoming very popular. Today there are easily more than 50, maybe more than 100 labs that study fly sleep. And it actually then was followed by the development of many other small models, animal models for sleep. And the ones that are being most for molecular studies are fish, zebrafish, roundworms, which is C. elegans, and Drosophila. But there are others, and they all bring some unique strengths to the table. So in octopus, for instance, there seems to be a stage of sleep that's like REM sleep, where they have an active sleep, just like REM sleep, and there is a skin patterning that occurs during that sleep that's similar to what happens during wake. Okay, so if you want to study REM sleep, the fly is probably not the right model, but maybe the octopus is. And so, sometimes when I talk about this, people will say, well, do you really think that, flies sleep like we do? And I think what I'd like to stress is we don't think that all aspects of sleep are conserved. Okay, we think that if sleep is as important as we all think it is, there must be some fundamental features of it that are conserved in the species that exhibit it. And those are the things that we are trying to get at. Okay, just want to keep track of time. Okay, can we use the fly to determine how sleep is controlled across the lifespan by different molecules? Young animals sleep a lot, and so do young flies. And so here, you're looking at day 1 flies compared to day 8 to 10. The light bars are day 1, dark is 8 to 10. For purposes of lifespan, 1 day in the life of a fly is equivalent to 1 human year, all right, because they live to 80 or 90 days. And so you're basically, looking at a 1-year-old human infant compared to 8 to 10, and this is sleep from a population of flies over a day-night cycle. So flies have most of their sleep at night, but they also have an afternoon siesta, and the young flies sleep much more. And you see that here also, that when you turn the light off or on, they fall asleep much faster, okay? The latency to sleep, they fall asleep much faster than the older flies. And I will come back to young sleep and what function it might serve later. But this is also to tell you that not only can you study sleep early in young animals where they have more, but also with age. So here are human subjects that have been monitored for activity, and you see in the young subject you have this very well consolidated activity during the day over a 5-day period. In the older subject, you see that this pattern falls apart. You start to get more daytime activity, and you start to get, you start to get more nighttime activity, and you start to get naps in the during the active phase, during the day. So you see the same thing in flies. Now, because of their shorter lifespan, you can monitor the same fly when she's young and when she's old. And I think I don't need to tell you that the sleep gets fragmented very much like it does in humans, where you now have more nighttime awakenings and daytime sleep. And we're interested in why this happens and in whether improving sleep would alleviate pathology associated with aging. So we can look at different parts of the lifespan, but what drives sleep? What molecules are driving sleep and wake in flies? When we first developed the fly model, we thought, we looked at the known neuromodulators, that neurotransmitters, which neurons use to communicate with each other, to see what effect they had. And I'll point to just two of them here, which are well known to affect sleep. GABA, which increases sleep, and it does so in flies and mammals. Dopamine, which decreases sleep because it promotes arousal. And again, that effect is conserved. So these things are so well conserved, in fact, that drugs that affect sleep in humans have similar effects on flies. And we've even used the fly to find new novel molecules that could affect sleep. But we didn't develop a fly model to say that what is true in the mammal is true in the fly. What you want to do is go beyond that. You want to use the unbiased approach, which was so successful for the circadian clockwork, and that is forward genetics. And we know Drosophila is a great model for that. You can screen lots of flies. It's well established for genetics. For our purposes, what was great was that the first circadian genes were found here, and they are, of course, the ones that time sleep. And as I mentioned, these genes are now even mutated in human circadian disorders. But I would argue that for sleep, there's an additional advantage of Drosophila, even beyond its advantage for circadian rhythms. And that is that circadian rhythms are very robust and very stereotyped. Okay? You see this nice 24-hour pattern, regardless of background and rearing conditions and diet. Sleep, on the other hand, is very variable. As you probably know, it's different from person to person, and in the same person, it can vary based upon what mattress you're sleeping on, what food you've had, and so forth. So if you want a mutant that you really want to use for molecular dissection, you need something that has a very strong phenotype. In other words, a very strong effect on sleep. And in flies, it's easier to get that because there's less compensation and redundancy. In mammals, many genes serve the same function, so you lose one and the others make up. In the fly, that doesn't happen as much. So we did a screen, and here is the first mutant we got from a screen. And I hope you can see here that these mutants, males and females, you'll notice here, by the way, the afternoon siesta is much greater in males than females. That's just the way it is. And these mutants have 80% less sleep than the controls. So here are mutants that have reduced sleep, isolated by us or by others. And we figured out what gene families they belong to. So the good news is that we can find genes that affect sleep, and they have conserved effects in mammals. So a lot of what's coming out of human studies of sleep now is implicating these same pathways. And so that's great. So we think that these are important modulators of daily sleep amount. But are these the drivers of sleep? Are these the things that go up and make you sleepy? The signaling increases, makes you sleepy. And the answer seems to be no, they're not. They're probably responding to some upstream signal that is driving sleep, and then they are making sleep occur. They're like the effectors. And so in these animals where you don't have these genes, they have reduced sleep, but they still have the need to sleep. That makes sense. Okay, they're like your classic insomniacs. They're genetic models for insomnia. So can we find something that actually drives sleep? And so we asked, can we increase sleep by increasing expression of any single gene? And this is work done in my lab by Hirofumi Toda, a former postdoc who now has his own lab in Japan. And he did a screen, and so this is the distribution of sleep across the different lines. We're interested in things that have increased sleep here. But it turns out that he got 7 lines, and which correspond to 6 genes, because 2 lines had the same gene overexpressed, increased in expression. And 5 of these genes, when you increase their expression, they reduce sleep. Only 1 increases sleep, okay? It's not easy to find sleep inducers, and we can talk about why that might be. This molecule turns out to be an antimicrobial peptide. We named it Nemori, which is the Japanese word for sleep, and it encodes a molecule that kills bacteria. Okay, it's an immune molecule. And what this molecule does is whenever there's high sleep need, which could be after prolonged wakefulness, or it could be in response to infection, because we all sleep more when we are sick, and so do flies. Then this molecule drives sleep. So what this molecule does is when sleep need is high, it drives sleep, and under conditions of infection, it is also killing microbes. So it's doing this dual function of helping the animal recover. And this has actually led to another project in our lab that we're very interested in, and again, we can talk about this later if anybody's interested. Is sleep during sickness the same as sleep following wakefulness? And I think the answer is no, but we're still working on this. Okay, I think I'm running a little bit late. Is it okay if I take 5 minutes? Yep, okay. All right, thank you. So anyway, so finally, can we use the function fly to determine function served by sleep? So why do young animals sleep so much? So this project was taken on in my lab by a physician scientist, Matt Kaiser, who now has his own lab. He's a very successful investigator. And as a psychiatrist, he was interested in the idea that early life sleep disruption might make an individual susceptible to psychiatric disorders later in life. And this, thinking about why young animals sleep so much, you know, dates back many years. So Bill Dement here, he's talking about REM sleep in particular, but he said it could be that it's required for development of the nervous system. And that is indeed what Matt found in my lab for flies, was that sleep in early life is required for the circuits, for the development of the brain regions that are driving adult behavior. And the adult behavior that he found was affected in particular is mating. So this is a behavior that's important for propagation of the species. And so flies have this very elaborate courtship ritual. I don't know if you can read the text there, but basically the male fly orients towards the female, he taps her, he flaps his wing, it's called like a song, and then he licks, and then he copulates. If you deprive flies of sleep early in life, so at day 2, and then assay them 3 days later at day 5, they show impaired courtship. This does not happen if you deprive them on day 9 and test them 3 days later, which is day 12. So the amount of courtship they do is reduced, but the completion of courtship is even more dramatically affected. They do not go through the whole process through copulation. And again, I'm going to show you a video, which was many years later. It's still poor quality, but it's better than the other one, I think. So here's a normal male. You can see chasing after the female, extending his wing. The one that's sleep deprived when young keeps losing it while he's courting. And he never quite makes it. And you see that these flies here are already, at the final step on the left. So this guy keeps, the one on the right he keeps trying to reinitiate it, and yeah. It's going to end pretty soon, but he will. Okay, there we go. All right, so this is in young animals. Why do we as adults need to sleep? And you'll hear a lot of reasons. And you just heard from Bob Stickgold about how you'll perform better and your cognitive ability will be better. And that is, in fact, a major outcome of sleep. And so we always say that the way to find out why a process is important is to knock it out, right? And see what happens. And if you knock sleep out, you have effects on cognitive impairment, memory lapses, as you just heard from Bob Stickgold. But you also have a lot of peripheral effects, like on immune system. You also heard about the risk of diabetes. A lot of this work on cognition has been done by my colleague and collaborator at Penn, David Dinges, and we've teamed up with him to look at what happens to molecules, metabolites in individuals that have less sleep, and we actually find exactly what Bob Stickgold said, which is 5 nights or 4 nights of 4 hours of sleep a night leads to a pre-diabetic condition. So, and the idea that cognition is affected by sleep loss goes way back. This is a paper from 1924 that shows that subjects that slept did much better than those that were kept awake at night. But we want to know on a very fundamental level, what is sleep doing that is helping these processes? Okay. And our mutants are implicating different processes, right? If you look at our mutants and what is affected in those mutants, there's effects on brain connectivity, on metabolism, brain waste, and immune function. And in my last 2 slides, I'm going to tell you that these are the 2 we are really focusing on. And we find that sleep is really important for these processes. Now, you will hear tomorrow from Maiken Nedergaard, who will tell you about her beautiful work showing how the glymphatic system is involved in waste clearance during sleep. The 2 processes that we work on are a little bit different, but I think they're all serving the same purpose. So during wake, your neurons are firing a lot in the brain. You're looking at the fly brain here. And there's a lot of metabolic waste building up within cells, but also just in between cells. And what we find is that during sleep, this waste is cleared. Within cells, it's cleared by a process called autophagy, and then in the brain as a whole, it's via endocytosis through the blood-brain barrier. And the other aspect of this clearing of the metabolic waste is that what it's doing is helping to maintain the integrity of neurons, in particular the mitochondria of neurons. So mitochondria are the energy workhorses of the cell. They are the ones that are generating the energy for neuronal firing. At the end of a day of wake, the mitochondria, I don't know if you can see that here, they get a little bit shattered. They build up oxidative damage. And we find that during sleep, this gets repaired. So my take-home messages are basically that we can study sleep at different stages of life, under different conditions. We found genes regulating sleep in flies. They have conserved functions in humans. And we believe sleep is important for metabolic balance, especially in the brain, but also the periphery. And that's my lab, awake and asleep. And I will stop there. Thank you.